So my name is Xuejian Wang, and I'm currently a third-year PhD student at Eawag. I'm very glad to have this chance to share with you some of my recent work and talk about the monitoring of cyanobacterial toxins. So the title of my presentation today is Tracking uh, Extensive Portfolios of Cyanotoxins in 5-year lake survey and identify indicator metabolites of cyanobacterial taxa. First, I would like to give you a brief background information, as some of you might not know much about cyanobacteria. So, cyanobacteria are regarded as the first living cells on Earth. Let me just get this. Um, yeah, there are many different shapes and colors of them, as you can see from the pictures. And under favorable environmental conditions, such as like high nutrient level and warm temperature, they could quickly reproduce and form blooms. So such blooms happened globally, including like in Baltic Sea, in China, in the US, and also in Switzerland. So these cyanobacterial blooms could cause adverse effect to the aquatic environment, such as like shedding light, consuming oxygen, restrict recreational activities, but also they could be harmful to our human health by constrained drinking water treatment. And then one major concern is this cyanobacteria could produce and release toxic secondary metabolites, or we just call it cyanotoxins. So the adverse effects of these cyanotoxins could include liver and kidney damage, tumor promotion, reduced DNA repair, and so on. And of all the cyanotoxins, 4 toxins received significant focus as the World Health Organization has set guideline values for each of them and the different scenarios. For example, the first one is called microcystin LR, which is the most well-studied cyanotoxins until now. And the concentration of it should not over 1 microgram per liter in drinking waters, and it should not over 24 microgram per liter in recreational waters. But actually, besides these 4 cyanotoxins, there are many more secondary metabolites co-produced by cyanobacteria. So our group, together with some collaborators, based on the published papers, collected the information of the secondary metabolites and made a database called CyanoMedDB, which contains over 2,000 structural information of the secondary metabolites from cyanobacteria. So based on their chemical structures, we could try to class all these cyanopeptides into all cyanobacterial metabolites into different classes. For example, the microcystins, which are also known as the most well-known toxic classes, but also could also include cyanopeptolins and the peptins and so on. And actually, until now, there are some reports say some of the metabolites from cyanopeptolins and then the peptins, they are bioactive. They could, for example, inhibit the enzyme activity, which requires we further evaluate whether they are toxic or not. But however, even there are so many other metabolites co-produced together with microcystins, but they receive less attention, as we can simply see from the publication numbers. So there are over 5,000 publications about microcystin, but when it comes to other metabolites, there's only to several hundred. And also we think if there are so many other metabolites co-produced at the same time, will microcystins always the most abundant toxins? So here's a chart. So each pie chart represents a lake, and the green color represents the concentration of microcystins. We could see in some lake, microcystin dominate the toxin profiles. But however, there are also many other lakes that are not dominated by microcystins. So in this case, we think it's also important to focus more secondary metabolites, not just microcystins. Now I would like to give you a brief introduction about our started target lake. So the lake called Greifensee, located in the north part of Switzerland, and it's a eutrophic lake, and it's a recreational lake, especially in the summertime. People go swimming there, but it also has cyanide blooms happen, as you can see from the picture. And I guess if you ever swim there, you could see the notice of blue-green algae in summertime. And in the middle of the lake, there's a monitoring platform uh, established by Eawag. And with this monitoring platform, we could get the information of some environmental parameters, such as the chlorophyll A concentration of the algae, the conductivity, and the oxygen in the water. So we conducted our sampling work near the monitoring platform from 2019 and 2023 with the help of our colleagues that I showed you before, the slide before. So we always took samples from the same location in triplicate, and then we did filtration to divide the sample into particle bound phase, or we can also call it biomass samples, and also the dissolved phase, or you could also call it aqueous or water samples. And through the 5 years we have 150 sampling days in total. So for the samples, we used the online SPE enrichment to enrich and purify the samples and measured with liquid chromatography coupled high-resolution mass spectrometry. So based on CyanoDB, the database, we used Skyline to interpret the data from the LCMS. In our lab, we have 18 standards that we could compare with our LCMS data, so we could calculate the absolute concentration of these metabolites or toxins. However, not just our lab, the commercially available standards for the cyanobacterial sector metabolites is always below the 1% of the compound in the whole database. So to also explore more other possible metabolites, I did suspect screening. So suspect screening, to simply put, I picked out some possible metabolites from CyanoDB based on different parameters of the LCMS data. And then I prioritized some metabolites based on their intensities, or we can say more or less the abundance of these metabolites. And then afterwards, I did the manual annotation with the MS2 spectra to confirm their structure. So go through all the process, I could see how confident I am with the metabolite that I defined. So the highest level, called level 1, which we have the reference standards, so we could confirm the structure and calculate the absolute concentration. The level 2 is probable structures, which means by checking their MS2 fragment, we could guess this probably very likely is the compound. So later, all the metabolites that I show you the results are within these two confidence levels. And in total, I defined around 35 metabolites and mainly belongs to three peptide compound classes, including anabaptins, microcystins, and the microgenins. So we could first look at the two target result. So in this plot, the blue and the dark color means the concentration in the water phase, blue, in biomass phase, black. And also the blanks between each year, that means it's mostly like late winter and early spring. So we didn't do the sampling work at that time. The black dash and the blue dash means the limit of quantification. Once the concentration below this limit of quantification, we don't really trust the absolute concentrations. So we can see generally for the two targets that show the peak concentration in late summer or in autumn seasons. The highest concentration for the variant MCRR is around 70 nanogram per liter, and the highest concentration for anabenapeptine A was around 100 nanogram per liter. If we remember the WHO standard for microcystin LR in recreational water phase, we could see the concentration here is way below 24 microgram per liter. And also the concentration of the cyanotoxins in this lake is also quite low compared with other, we cannot say popular cyanobacterial bloom lakes in the world. However, with this data, we could still learn the dynamics of the toxins. So here we could see in some years, for example, 2019 here, the blue color is a bit higher than the black one. And also in 2022, and also in 2023 for anabenapeptine A. Actually, these are the exceptions. Normally, the concentration is way higher in the biomass samples. That's why after this slide, I will only show the result of the biomass phase to you. So here I show the peak area range of all the metabolites that I defined in a descending order of median values. The peak area is the LC-MS response. Here we don't use peak area to show the absolute abundance because of the instrument. However, the higher the peak area, that means the easier we could detect with our instrument, and it's easier for us to do the monitor analysis. We can see the two compounds that I showed you before. They're high enough to be detected by LC-MS, but still they're not the highest, not the easiest compound to be detected in a lake. If we colored all the metabolites based on their compound class, we could see the blue and the yellow color means anabenapeptines and microgenins are way abundant in the case of LC-MS response, which means they are easier to be detected with our instrument. I also further calculate the detection frequency of each metabolite. So it was calculated by dividing the dates that the metabolites was detected by the total sampling date. So if we put in together the frequency information and the LC-MS response, you could see there are some metabolites, for example, the left part here, they are highly frequent detected. And also they have high LC-MS response. And also on the other side, some metabolites, they are frequently detected, but the detection, they have low LC-MS response. There's a type of some metabolites, they have low frequency, but then they have different response on the LC-MS part. And then if we calculate the detection frequency by the compound class, we could see that for anabenapeptines and the microgenins, they will detect for 90% of the sampling time we have, which is higher than the microcystins. So if we put together the absolute concentration, the LC-MS response and the detection frequency, we could see that in our lake, microcystin, the most popular one, seems not dominant the lake when the bloom happens. And besides the samples we took in the lake, there were also 3 cyanobacterial strains were isolated from the lake when bloom happens. 2 of them were isolated during the sampling time in 2020, and 1 was isolated earlier in 2011. So by the microscopic check and the 16S RNA sequencing result, 2 of them I confirmed as microcystis, and 1 is Planktothrix. Since we have them isolated, we have their cultures in the lab, So I could measure what type of metabolites they could produce and try to see whether the metabolites they produce we could also find in the lake. So here are the metabolites that each of the strain could produce. So first we could see that some metabolites they could produce in common. For example, the alkylamide Y and the beta-peptine F and the beta-peptine B. But also there are some metabolites that are only produced by one of the strain. For example, all the metabolites they are in the colored boxes. Then when look at this special metabolites, I was thinking whether I can pick one metabolite for only one strain to somehow indicate this isolate. So I picked them based on the compound in the lake, their detection frequency, and then their LC-MS response. So I also picked oxalamide Y because it could be produced by all the three strains, which should represent cyanobacteria in general. If we look at the time series of all the four indicated metabolites, we could have similar conclusion as we had before with the absolute concentrations. In late summer or autumn time, they could reach their highest concentration, but the time they reach their highest concentration is different. For example, for the blue one in 2020, it reached the highest concentration or abundance in August, while the other two reached in October. And also in 2023, the three metabolites reached the highest concentration abundance in different months. So by they reach the highest abundance, we could think maybe that means the producing cyanobacteria, their biomass also increase highly in that month, or the productivity of the producing strains is very high in that month. So it's in a way, the indicated metabolites reflect the cyanobacteria strain. So putting all the information together, here comes to the conclusion. So in our target lake, I defined 35 metabolites, and microcystin, the concentration was always below the WHO standards. And we could conclude that in this lake, it seems that N-beta-peptins is the most abundant and the frequent metabolite. And by analyzing the toxins produced by three strains, we could find the indicated metabolites for each strain. And by following the changing patterns of the indicated metabolites, we could reflect the variance of the producing strains. And it also can be used in other lakes, especially if the lake there are many different cyanobacteria bloom happens. With that, I would like to thank my supervisor and our collaborators, and thank you all for listening.